Today, I'm going to walk us through the SWAMP MQOs, the measurement quality objectives that we use in SWAMP to ensure that data quality is standard and sufficient to be released to the public. And we can all get along with our data, analyze things on an equal footing. And then I'm going to also look at the recent changes that we've made to the SWAMP MQOs and why that was. So really the objectives of our MQOs in SWAMP is first data integrity and quality, right? We want data that's produced under appropriate conditions, proper controls, data conformity. Data is always held to the same standards and reported in a uniform manner so that people can compare data. The public, who we are stewards for data for, can look at the data that's produced in their home state and compare it themselves. And also that data is legally defensible. We want to make sure that the data that we are generating is within the limits of the methods, and data of suspect integrity and quality is appropriately flagged, and ensure that legal and programmatic decisions aren't based on inferior quality data. We need to have a really rigorous system to ensure that our decision making is based on foundational data. So I'm going to walk through some of the MQOs. This is for indicator bacteria in freshwater. Obviously, we have other MQOs in development for other methods, and this largely focuses on two of the most common methods for measuring bacteria, and that is plate-based methods, so filter and plating, and multiple tube fermentation style methods, which is IDEX or just the traditional NPN tube-based methods. So first, we have sterility checks. So any lab that's going to be running these analyses and generating data that is SWAMP comparable, you have to run your sterility checks. And the first is that, per new lot of dehydrated culture media, if you're using a dehydrated culture media, you have to ensure that there's no growth, right? From the onset, just the evaluation of the bias from contamination, and then also non-sterile filters and pads for every lot should be tested. These are just general laboratory things that should be happening in the laboratory. We don't require these to be reported. I don't want to see everyone's results from their lots that they're testing for their media and their filters. That would drive me insane. And then so for membrane filter methods, again, no growth on the media, filters, buffered dilution water, rinse waters. And then in the multiple tube fermentation, same thing. You're looking through your media and dilution waters. Now, ensuring there's no growth in those. Then we get to positive and negative controls, laboratory positive controls, laboratory negative controls. So every time you use a new lot of dehydrated culture media, or you're using a new lot of commercially prepared culture media ampules, and then per batch of laboratory prepared culture media. So every time you make a batch of culture media in the lab, every batch you should be running a test on that as well. So for both positive and negative responses. Now, laboratory blanks. Sometimes I see people conflating laboratory blanks and negative controls together, and that should be remedied. Laboratory blanks are not negative controls, right? Negative controls are typically an active organism, just not the organism target, not something that the media or method is designed to target. So laboratory blanks are only necessary to be reported when a dilution is made in the laboratory. So if you are diluting across different sensitivities, you do need to report a laboratory blank to ensure your lab is competent and capable of not introducing processing contamination. And then for fuel blanks and equipment blanks, we don't have any hard and fast requirements for fuel and equipment blanks, but that's just per your method or SOP. We rely on those. Now I want to get into how things have changed recently, and that is due to precision. So laboratory duplicates for bacteria, and this is where things get a little bit dicey. We've been using kind of the same metrics for precision for a little over a decade, and that was a method called R-log, or 3.27R. The basis for this was this is how it's outlined in standard methods, and standard methods were largely developed utilizing plate methods. So, and because plate methods have been around longer, I guess, and they were standardized first. So the RLog method was generated for plate style methods, and calculating that and reporting that is what has been the norm. Now, I had a fun conversation with the authors of standard methods, and I'll explain that in a little bit, but this is how you calculate RLog. It is, you take your duplicate samples, you run a sample in duplicate, take the logarithms, find the range of those logarithms, and then you find the average of those differences. And you do that for the past 15 laboratory batches, and that gives you a target. And based on that target, you know your lab's threshold for precision. And if you are to run the next set of laboratory duplicates and it exceeds that precision value, then you'll know that you're out of compliance. Your precision is not appropriate, and we'll need to qualify that data. Now, we were applying this method for both plates and IDEX style methods, so MPN and MTF style methods. Now, I talked to the people that wrote the standard methods, and I was assured on both accounts that this is a pretty common conflation, but this calculation doesn't apply to multiple tube fermentation methods or any of the IDEX color quantity tray style methods. And I'll get into why that is. But regardless of that, the standard methods ongoing precision demonstrations are still outlined in the general laboratory QC for the standard methods. They're still considered standard lab practices. So matrix duplicates are in there, but the thresholds haven't been really determined. So we decided that we needed to generate some. So first, we need to understand the differences in reporting. Mostly our regulatory limits, everything you see, even like the bacteria provisions, things like that, metrics are all, generally they say CFUs, right? We're talking 100 CFUs per 100 mil sample or what have you. Now, MTF, MPN, most probable number units, multiple tube fermentation style methods, generate data in an MPN format. And that is, again, it's a probability distribution. And we are reporting what we consider the index value. And that index value is the most probable value for that sample. But every one of these values has an associated confidence interval with it. See, the CFU values are discrete values, right? You get a plate, you count the numbers on it. There's a small amount of variation between individuals in the lab. One might count 36, one might count 37. And there are ways to quantify that. But for the most part, CFUs are discrete and MPNs are probability distributions. So granted that, how do we come up with a value to ensure that when we run a laboratory duplicate, that we are within reasonable bounds for precision? So the first thing we entertained was that, well, we can just look at it based on our statistical evaluation. So if the confidence intervals don't overlap, then they are separate, and then that's not good enough. The laboratory duplicates are then not precise enough. The problem with that is there's a lot of gray area, and that's not a very good metric for precision. When you're talking about, I'm running the same exact sample twice, granted there are bacteria values are a little bit more not as stringent as maybe a chemical value. So when they're separate, obviously it's not a good enough precision. We wanted to get a little bit more in depth than that. And you can see for one sample that has an index value of 100, it would take a separate sample, running the same sample would have to give you an index value of 153 before they were considered not precise enough. Now, again, that's not as well enough for us, and I'll get into how we calculated that. So what we decided to go forward with is using the MPN, that index value, as the target metric. So really this confidence interval says that 95% of the time, if you run this sample 100 times, your MPN should fall within these confidence intervals. Now, it would be terrible if we ran the sample one more time right after that, and then the MPN was directly outside of that confidence interval. Again, not a lot of satisfaction in our actual confidence interval. So for this instance, these confidence intervals are dynamic. So throughout these calculation tables, the relative percent difference between one value and another varies quite greatly. It can vary from 20% all the way up to 60% RPD, which is acceptable depending on the confidence of these tests. So this is what we have gone with. If you run a duplicate sample and the confidence interval is outside of that first sample's confidence interval, the index value is outside of the confidence interval of the first, then we would consider that not precise value, and then we could qualify that appropriately. So in order to look at this in context, we looked at about 15 years of laboratory batches, and that's a little over 1,000 lab duplicates from a variety of our labs at the state water boards. And we see when the MPN index value exceeds the confidence interval, we look at what the RPD distribution of that is. And this is the distribution I mentioned previously. Now this exceedance can occur as low as 20% RPD, and can also occur all the way up, 100, 120%. It depends. So we can't set a fixed number on what is considered precise enough, and that's why an RPD value isn't appropriate. So we needed a dynamic number. Now we tried to apply the R log. So R log is again what we've been using for a little over a decade, and we tried to apply this R log metric to all this data, and what we found is that R log is only successfully flagging batches about 20% of the time that they exceed. So that didn't give us very much confidence, and it's only flagging batches. It only starts to flag batches at RPD about 50%, with a mean of around 90 to 100% RPD. Again, when you're talking to the public, or if you are going to be, again, shutting down a beach based on a value, you want to be able to say, yes, if someone asks you if you can repeat that measure on the same value and you get something completely out of the confidence interval, again, I think that leaves us open for legal interpretation. So this is the metric that we've went with. I have a narrative written for this, along with the rest of the data analysis, if you'd like to see it. But again, this is what we'll be moving forward as of this month. The MPN for laboratory duplicates for the multiple tube fermentation, most probable number units, need to be within the confidence interval of the duplicates. Now, before I get into anything else, I wanted to make a small announcement. The California Water Boards and the California Stormwater Quality Association will be co-hosting a 3-day statewide summit on bacteria. And the goals here are going to be to identify the priority technical and regulatory actions needed to recreate safely in California's rivers and ocean waters and to eat shellfish safely. This will take place September 14th through the 16th, tentatively set for the Cal EPA building in Sacramento, pending applicable restrictions. So we invite everyone to save the date. Katie's on this call as well, and she's on that board setting this up with us as well. So with that, if you'd like to visit the Swamp IQ microbiology webpage where we have these MQOs posted, that among other things, or if you want to contact me directly, I'm always happy to talk about them. Other than that, any questions, I'd be happy to field.
Thank you, Charles. If you have a question, please go ahead and raise your hand. Send it in via the chat, whichever makes you feel most comfortable.
Nope.
Hi, great presentation, and it's very relevant to us because obviously we presented an alternate method to MPN and to membrane filtration, and we have good side-by-side, validation with both. However, there is one question that is interesting, I think, to address in the context of your presentation, and I'd like to know your opinion. Neither MPN or membrane filtration methods can measure particles, bacteria that are attached to particles. So let's say you have, a fecal particle there that has thousands of bacteria and it doesn't get homogenized. So this one will end up on your membrane and will form a single colony, or it will end up in a single well of the, of the colilert or any other multiple tube fermentation method that you use. So it will be counted as a single bacterium, whereas, if somebody swims and, drinks that water, well, he will have a huge charge of bacteria and pathogens associated with it. So our method measures all the bacteria in the sample, including the ones on the particles, because it cultures the whole sample and measures on the whole sample. And so it does measure those bacteria on particles. Sometimes we see differences as high as a factor of 10 if we look at wastewater effluent between, filtered and unfiltered samples measured the same way. We can measure the difference, and basically the standard method only measures 10% of the bacteria present in some type of water like that. So I was wondering, how is that taken into account in the regulations, or is it a subject to look at in future, changes to the regulations?
Yeah, I think obviously there's always room for improvement, and these are limitations of the methods. Actually, when you were giving your presentation, that was one of the follow-up questions I was going to have. We were getting kind of in the weeds. What units are you producing in? Well, looks like fluorescent units, but you're actually calculating actual number from. But yeah, these are all limitations of the methods that we currently have in using culture-based MPN probability metrics. So it's almost like we can never have enough data to really get at it. Can we factor in turbidity to try to massage some of that data to get a better, more accurate number? Possibly, but to get that through the pipeline and get something validated that would incorporate that, I haven't seen anything, and I don't see anything under development. So I would be interested if someone has some ideas, because these are the methods that we have set up for SWAMP. We have MQOs for, because they're so universally distributed. So we want to make sure that the data that is produced is congruent. But with new technology, we are, I'd love to be on the ground floor talking about how we can make sure that data is comparable.
Great. I would love to engage a conversation. I'll follow up by an email. Thank you so much for the great presentation.
Of course.
Thanks again, Charles.